Good morning, everybody. Or is it good day? It's good day, ne? Good day, everyone. My name is Tutuzire Kupega. My email address is dkupega2204@gmail.com. So today, remember yesterday we had to finish up with that other exercise and we didn't get to do it. So now we are going to go straight to that exercise. We answer it, and then as soon as we are done with that, we move on to revision or preparation of the assessment. So what I prepared for today is that we cover some past papers together. So we'll go through some past paper questions. So the assessment will be set up like a June exam. Ne? So I want us to cover, like, just get to understand how questions are asked, how you can answer them, just so you can get the feel. So we'll do some past papers, answer it together, and just prepare for the assessment. Ne? So the exercise, though, the one we are starting with now to finish up with our unit for the topic of homeostatic control in kidneys, is, let me quickly go to that slide because we were supposed to cover it yesterday. There is that slide of the exercise. Remember, we covered all of this. Now there's the exercise. Extracted from Via Africa Life Sciences Learner's Book. This is where the exercise I extracted it from. Question 1 goes, tabulate the different processes of the kidney. Do you remember the processes of the kidney? We covered, I'll only show 3 here. Why? It's 15 marks, ne? So it will be like 5 marks per process. But you remember when we did it, we had like 5 processes. But right now I have covered the 3 now. You have ultrafiltration, there is reabsorption, and there's tubular excretion. But we know there are more than 3, right? We did the, let's see them on this diagram. We had more than 3. We have reabsorption, we have ultrafiltration, we have tubular secretion, we have tubular excretion, and then we have urine formation, all 5. So you can really explain any of the 5. Here I've just showed you the 3, but you are welcome to explain any 3 from the 5. What guided me here is the mark allocation. So what happens during the ultrafiltration? This is revision, guys. Remember, now we are just marking. We've covered this work. So I think it's clearer now. And if it's not, you are still most welcome to tell me. And I will go take you through the work if you're struggling with certain portions here. Ultrafiltration, the afferent arteriole. The diameter of the afferent arteriole is wide. Remember, the diameter of the afferent arteriole is wide. This here allows blood to enter the glomerulus. Blood pressure in the glomerulus is very high. Why? Why is blood pressure in the glomerulus very high? Tell me the answer. I remember I explained why the blood pressure is high. Who remembers that explanation? Who remembers why? I explained why the blood pressure in the glomerulus is very high. Who remembers that explanation? It has to do with the afferent and efferent arteriole. So who remembers that? Okay, should I just give it to you since you are not going to tell me? Okay, let me tell you then. Oh, there's someone in the chat. This separates waste from blood proteins and corpuscles. Okay, I hear that. I get that. That is correct. But explain to me why. Yes, it's when the blood pressure is because of that separation, yes, when it filters. There are pores in very thin squamous layer. No, the blood pressure. The reason for this high blood pressure, guys, is because of the afferent arterioles and efferent arteriole. You remember the afferent has a wide diameter and the efferent has a narrow diameter. Let me try and draw it for you so it can be clearer here. So it's like this. Let's just try and draw it like this. So the afferent would be like this. It would be wide. And then it would get smaller. You understand? Because it gets smaller here. Here at the afferent, it's wider, meaning there's more volume, right? But now, when it's going to the efferent, which is the narrower arteriole, it's narrow in diameter. This is the efferent. Remember, I said, remember AE. Efferent is narrower. So because here, why it's narrower, meaning the volume is decreasing. Now this change in volume here, you had lots and lots of blood entering here with the large volume. Now it needs to pass through this very smaller, narrower one now. The blood pressure is increasing. You are decreasing the volume, thereby increasing the pressure. Do you understand how it works? Indirectly proportional. Pressure and volume are indirectly proportional to each other. We are decreasing the volume here, thereby increasing the pressure. And that is why the blood pressure here will be very high. Do you understand? So whenever you get, we even did an exercise where we covered this topic about blood pressure in the glomerulus. Whenever you get asked this question again, remember it's because of the diameter of the afferent and the efferent arterioles. That is the reason. And then now, moving on to that part here, which we're explaining, the pores in the squamous layer of the capillaries and the podocytes in the Bowman's capsule allow blood to filter rapidly into the capsular space. So it's filtering rapidly because of these pores, yes, and the increased blood pressure. So Buhle had a very good idea when she mentioned it's because of the pores. So remember, you see with the pores now, they are now smaller in diameter than the efferent. So the sizes, they play a huge role. That is why it happens rapidly. It's first because of the afferent's wide diameter and the efferent's narrow diameter. And then now, when it moves to the pores are also very, very small compared to the efferent arteriole. And that's why now, what would happen? The filtration will occur rapidly into that capsular space. Then the filtrate will consist of small particles of water, glucose, amino acids, vitamins, mineral salts, urea, uric acid, and creatinine. The filtrate is what has been filtered from the blood. So these are the substances that will be removed from the blood. It is the filtrate. The filtrate, it would be what? Particles of water, glucose, amino acids, vitamins, mineral salts, urea, uric acid, and creatinine. So whenever you're asked what can you find in the filtrate, you'd find different things in the filtrate, which will include these ones over here, right? The large particles, which are mainly blood proteins and blood corpuscles, will remain in the capillaries of the glomerulus. So what will be filtered is this, but in the capillaries of the glomerulus will be the blood proteins and corpuscles because they are large particles. So that's the process of ultrafiltration. The next one after that is the reabsorption. As the filtrate travels along the nephron, useful molecules that are in the filtrate. So these useful molecules are what? Glucose, amino acids, and water. You see here, in the filtrate, it includes various others as well. There's also mineral salts, there's urea, uric acid, and creatinine. But urea will just be going to be excreted, so it's not going to be reabsorbed. Uric acid will not be reabsorbed. Creatinine will not be reabsorbed. So the useful ones, the ones that are required by the body at that particular time, would include maybe glucose, amino acids, and water, will be reabsorbed into the blood. Where is the blood? The blood is in the capillaries of the glomerulus. Remember, the blood remained there. In that sac-like structure, they remained in the capillaries of the glomerulus. The process here, the process of reabsorption, will be either active or passive. Remember when I mentioned to you active or passive processes. Passive one include diffusion and osmosis, but active one will be that of active transport. Active transport requires the input of energy in the form of ATP. That is why it is called active. Active transportation means you require an energy input. And the name gives it away here, active transport, meaning with this form of transport, you require an energy input. And how does that work? It moves the other two, which is osmosis and diffusion. In osmosis, it would be the movement of water from a high water potential to a low water potential, right? That is osmosis. And we've seen there when ADH and regulates the water concentration, it happens, all those processes of reabsorption occur here, where ADH is present here in this stage of reabsorption, whereby now it will make the tubes more, it'll ensure that the tubes are more permeable to the water as it moves into the capillaries and into the capsular space. So it keeps on moving there to balance things out. But if it's not the water moving, remember water will move through the process of osmosis. But if it's the movement of glucose or the movement of amino acids, then that happens through the process called diffusion, because diffusion is the movement of molecules or substances from a region of high concentration to a region of low concentration. So that's the difference. The only difference, right? It's the same principles. The difference is in osmosis, it's the water that moves. But in diffusion, it's the molecules or substances that move. Yeah? Then, for active transport, why do you require an energy input? Because active transport is actually going against the concentration gradient, meaning you would move things along from a low concentration region to a high concentration region, whereas the other ones would move from a high concentration to a low concentration. So active transport you'd normally see with the movement of potassium ions or sodium ions. Those are the ones that usually move against the concentration gradient, meaning they would move using active transport. So remember that part. It will make more sense the more you understand those concepts. Then what happens after? The cuboidal cells. Cuboidal cells have, these are the adaptations to ensure they function better and efficiently. So those adaptations include what? The tubules, the tubules have adapted to include what? The cuboidal cells, which have a rich supply of mitochondria to provide energy. And I explained why. Because in the mitochondria, which is known as the powerhouse of the cell, that is where cellular respiration takes place. In the process of cellular respiration, the input is carbon dioxide and your glucose molecule. Then the output would be oxygen, water, and carbon dioxide. No, man, my bad. The input is oxygen and glucose, but your products, your byproducts would be water. It would be carbon dioxide and energy. So that is the site of energy synthesis. So that's why they have these rich cells which have the rich supply of mitochondria, because you will need this ATP produced in the mitochondria through cellular respiration to what? To assist in active transport there. You understand? Then, yes, for active reabsorption, as it's saying, it assists in active transport. Then, what else does it have? It has microvilli, which increases the surface area for absorption. Microvilli increases surface area for absorption. Then we have water, Water is reabsorbed by what? By the distal convoluted tubule, where aldosterone will regulate the reabsorption of water. Of salts, man, my bad. Aldosterone is involved in the regulation of salt, but ADH is involved in the regulation of water. Then next one is tubular excretion. In tubular excretion, the potassium ions were completely reabsorbed in the proximal convoluted tubule, and they are then secreted into the distal convoluted tubule. This secretion is enhanced by the hormone called aldosterone. Hydrogen ions are also secreted into the distal convoluted tubules, causing the urine to have an acidic pH. You see, hydrogen ions contribute to the acidity of the urine. And then from the distal convoluted tubule, it would move down until it gets to the collecting ducts, where it's now on its way out of the kidney. So remember these ones, this was just exercise, guys. You can go through the videos, you can go through the books to understand some more. And I'm here, like I said, I'm always here. Send me an email if you need my explanation even more, or if you're doing any work, and I'll gladly assist. Question 2. Tabulate the role of the two hormones involved in the homeostatic control of the kidney. So before I show you the answer there, tell me anything you know about the hormones involved in homeostatic control of the kidney. You can start by telling me their names, and then you can move on to telling me anything at all that you know about these hormones. You can type in the chat box, or you can raise your hand and we'll give you a chance to speak. Either option, but just tell me. We covered this yesterday, so it's still fresh in your memory. Which are those hormones, and then what exactly do they do? We even covered some just now, been explaining. So tell me anything you remember, the names of these hormones and their roles. I'm gonna wait for your answer. I'll give you a few more seconds. It's 12:18 now, so I'll give you 1 minute. As soon as it hits 12:19, I'm gonna continue. I'm hoping you're quiet because you're typing, ne? Okay, guys, it's 12:19. I don't know, you're not talking to me today. All right, I'll just go on to the answer. The two hormones are ADH and aldosterone. Remember that. Please remember them, ne? If you had forgotten them, remember them. ADH, first up, it regulates the water content of the blood, as well as the body's temperature. Remember when Kuzwayo asked the question about fat as well, was when we were discussing this part of ADH regulating the process of sweat. But in the kidney particularly, it is involved in the regulating water content of the blood, as well as the water content of the blood in the kidney. Then in other processes, it can regulate sweat. But now we are discussing the kidney. So that's why we'll only focus on the ADH's functioning in regulating water content. So what happens? A decrease in the water content, which is the water potential, of the blood due to excessive sweating or insufficient water in the diet would cause the osmoreceptors in the hypothalamus of the brain to stimulate the posterior lobe of the pituitary gland. Remember what the pituitary gland is also called? It's also called the master gland. Why? It is involved in the processes of all other glands. Most of the other glands, actually, not all, just most of them. It's another name for pituitary gland is hypophysis. So it stimulates it to what? To release ADH. So ADH is released from the pituitary gland, which was stimulated by the hypothalamus. And the hypothalamus was alerted by osmoreceptors. What did the osmoreceptors actually find? They found that the water content in the blood was too low. So that's how it works. Then absorption of water from the distal convoluted tubule and collecting ducts will be regulated by ADH. Because what will ADH do? It will ensure that it controls the permeability of the walls of the nephron and those associated capillary tubules. Then what happens? ADH will increase the permeability of the walls of the distal convoluted tubules and the collecting duct, which will then cause water to be absorbed into the tissue fluid and the blood, which will then produce the concentrated urine. Because now it's moving, water will be moving out from the filtrate and would be moving back into the blood, thereby leaving the urine to be more concentrated. You understand? The urine will now be more concentrated because it will be losing water to the blood. So the water will move from the filtrate, as it was filtered during ultrafiltration. It will now move from the filtrate to the blood, whereby now it will leave the urine to be concentrated. And that's okay, because right now you actually need to excrete that. You can't excrete what the body needs. The body at that present moment needs water. So it needs to take that water from the filtrate back into the blood. So the urine will be concentrated. Other word is hypertonic. So when we say hypertonic, we actually mean concentrated. What is this process known as? It's known as antidiuresis. And that is why ADH is called the antidiuretic hormone, right? Because it is involved in the process of antidiuresis. Then, the opposite now, when a high water content of the blood decreases, a high water content of the blood is there, it's present. What happens? It will decrease or inhibit the secretion of ADH. So this process, you'll see it in negative feedback regulation. When it sees now that no, there's now too much water content in the blood, then the secretion of ADH will be stopped. How? The osmoreceptors again will alert that, and then it will alert the hypothalamus to stop stimulating the pituitary gland so that the production of ADH can be inhibited or stopped at that particular time. Then that would cause the permeability of the distal convoluted tubule and the collecting duct to decrease. Then large amounts of dilute urine now will be excreted. Why? Because now the water concentration would be high in the urine. So when it's high, it is called what? It's a dilute solution of urine now because there's more water. This one here is known as diuresis. So the words here, these ones should guide you. Diuresis and antidiuresis. Antidiuresis means the antidiuretic hormone is present, meaning ADH. But in diuresis, it means the ADH is not involved here. Then, now we move on to the process of aldosterone. Aldosterone, what is it doing here? Aldosterone is responsible now for the control of the salt content and pH of the blood. This is secreted by the cortex of the adrenal gland. Remember, I even showed you the diagram of the adrenal gland and where it is located on the kidneys. Let's quickly go and show and see it. There. This is the kidneys. Those are the adrenal glands there. There's the right one and the left adrenal gland. Then you see the cortex. The cortex is the outer layer, this one in yellow. That is the cortex. And then it's on the outer layer. Inside, it's the medulla. So cortex is secreted, the aldosterone is secreted on the cortex, which is the outer layer of the adrenal gland. Then when there is a decrease in blood volume, why would there be a decrease in blood volume? That would be a result of a loss of sodium ions. So when the blood loses sodium ions, the blood volume will decrease. Aldosterone will then stimulate the uptake of sodium from the nephric, which is the tubular filtrate, into the blood. Remember, the filtrate has all of these substances. So they will be taken from the filtrate back into the blood. For example, this is a typical example. You threw things out and you didn't pay attention. Maybe you're throwing out some papers, lots and lots of papers. And when now you're looking for your homework, you realize you might have thrown it out with all the other trash. So you go back to the rubbish bin, you look through it to find your homework. That's basically what's happening here. When the blood realizes there's not enough sodium or there's not enough water, it goes back to the filtrate, which is the rubbish bin in this case, to look through it and find what it needs, take it back to where it is needed. So that's what happens. If you were to throw things out and you realize you threw out money, would you leave it just because it went to the trash? Or would you go search for your money? You would go to the trash bin, right? Look for your money, because whether it's in the trash or not, it is money and you would want it back. Stimulate that uptake of sodium from the nephric filtrate into the blood, mainly from the distal convoluted tubule. Then the uptake of sodium ions results in the reabsorption of water into the blood capillaries in the kidney. So the more the sodium ions are being uptaken back into the blood, then it will also result in the reabsorption of water as well. So these two, they work together, meaning aldosterone in the regulation of salt will also be working together with ADH, as the water now will also be taken in. The kidneys help to maintain the salt content and pH of the blood by the secretion of hydrogen ions from the distal convoluted tubule. Remember, the distal convoluted tubule is that last one there before it goes to the collecting duct. Let's go see that one quickly. There, distal convoluted tubule is this one here. You see the proximal one is that one closer to the glomerulus or the Malpighian body. Then the distal convoluted tubule is that one on the other end now. Then all that thing, all those filtrate that is left now, all the filtrate that is left in the distal convoluted tubule after the reabsorption process is done, will then move on through the process of urine formation, where it will going to be collected by the collecting tubule or collecting duct, and then it moves to the ureter where it will be excreted. So it will cause what? Because the hydrogen ions will be secreted into this distal convoluted tubule, the urine then will have an acidic pH. Because when you add more hydrogen ions, you are making it more acidic. You understand? So yep, that is that part that I wanted to do with you. Now we are done with that exercise, guys. So like I said, we will do our revision immediately after this exercise. So do you have any questions before we start with the revision? Okay, if you have no questions, I have questions for you and I need you to interact now. This is from the June 2017 common test from the Department of KZN. Then now it's the questions. Let us work together. These are typical questions you are most likely to find in your assessment. Section A, question 1. You see the instruction here. It says 1.1. Various options are provided as possible answers to the following questions. Choose the correct answer and write only the letter A to D next to question number 1.1.1 to 1.1.10 in your answer book. For example, if in 1.1.11 the answer is D, that's how you'd write it like that. So you don't have to write the whole option. So let's say 1.1.1. The answer is gametophytes and gametophytes. You won't have to write that. You just have to write 1.1.1D. So this makes it easier for you as well. You don't have to waste time writing unnecessary things that are not required. If the question is clear, just write only the letter. I mean, save your time because it's a long paper and the time allocation is very specific to the mark allocation. So you don't want to waste your time on things that won't even give you marks. Please remember that. And then question 1.1.1, I need you to be very active in the chat box now. Question 1.1.1, the dominant generation of the mosses and the angiosperms respectively is what? Thank you, Buhle. She's giving me the answer and she says it's A. The most dominant generation of the mosses and the angiosperms respectively. Understand respectively means in that order they are written in, right? So if they say mosses and the answer is gametophytes, it means you are saying mosses, the dominant generation is gametophytes, and in angiosperms, the dominant generation is sporophytes, right? That's basically what I'm trying to say. We need to also pay attention to how the question is phrased. So yep, the answer is A, which is what? Gametophytes and sporophytes. Meaning mosses, the dominant generation is gametophytes, whereas in angiosperms, the dominant generation is sporophytes. Thank you, guys. Next question, question B. The diagram below represents the reproductive structure of a plant group that you have studied. So which plant group has the above reproductive structure? Butle has answered already. It is the pteridiophytes. Pteridiophytes. What do you think it is? Let's see. Good, Butle. Wow, Butle, did you study? Have you done this paper? Or you really just know your work, ne? I like this. I love this, actually. And then now 1.1.3. Bacteria that convert light energy to chemical potential energy stored in organic compounds are called? Good, Buhle. Those ones are called autotrophic bacteria. Why? Auto is telling you. These ones are producers. They produce their own food here. Meaning they convert what? They convert light energy to chemical potential energy stored in organic compounds. So this is a process you'd usually see in plants and you hardly see in bacteria. But now we have learned that some bacteria are actually autotrophic in that they can make their own food. They convert inorganic material to organic material. That is how great this is. And then question 1.1.4, study the list of characteristics below. We have coulomate and triploblastic, acoulomate and asymmetrical, diploblastic, bilateral system, radially symmetrical. Which one of the following applies to the cnidarians? Which one there applies to cnidarians? I think I should do this more often, guys. I like how you interact when we do this. Like, I haven't seen you guys this active, like both of you at the same time. The answer is B. 1.1.4, the answer is B. I think the next session, when we do this, we should do Kahoots. Do you know Kahoots? Can you both answer me if you know Kahoots, are familiar with it? I'd like us to try it. I see that you interact better when we ask questions and you have to just provide an answer. So with Kahoots, it will be typically the same thing. It's like a game. You'd log on to kahoot.it and then I'd be asking different questions and you just select the correct answer. Or in certain cases, you need to type in the correct answer. Are you familiar with Kahoots? Have you done it before? I'd like to try it with you guys because I see how you interact better when it's questions such as these ones. So do let me know, ne? And then we can try it. And then now, the next, I was sorry, I didn't I explained this properly. Cnidarians. It is three there, it's low. Let me highlight those ones as well. Cnidarians, it's what? It's diploplastic, or diploplastic, and V, which is radially symmetrical. So those are they apply to the cnidarians. Those are the features you'd find in cnidarians. And then next question is, I didn't do it nice. Okay, next question is, the grade 11 learners conducted an investigation to compare the effect of 3 antibiotics of the growth of the bacteria. They set up agar plates containing nutrients and bacteria. They set up agar plates containing nutrients and bacteria. Sterile discs 1, 2, and 3 containing 3 different antibiotics were then placed in each of the plates. The dots represent the bacteria. So you see the dots there. The results of this investigation are represented in the diagram below. So antibiotic 1, antibiotic 2, antibiotic 3. From the above, it can be concluded that the bacteria are least resistant to, okay, Kuzwayo is, okay, I've got 2 different answers there. Kuzwayo is saying A and Butle is saying C. Let us look at these things and analyze. Let's look at antibiotic 1. Look at this agar plate here, and they've put antibiotic 1 over here. But you see there are a lot of dots there. There are a lot of dots, meaning now these are the bacteria there. This is the antibiotic, this is bacteria. And number 2, there are a lot more dots on number 2 than there are on number 1. You understand? And then number 3, there are less dots than on any other ones. So this bacteria here is least resistant to antibiotic 3. So the correct answer is indeed C. Antibiotic here is least resistant to, the bacteria is least resistant to antibiotic 3. And you can see that with the number of bacteria that remain after you put the antibiotic. It works on similar concept, guys, to when you use soaps, different soaps, or different disinfectants to clean your surfaces. And that is why this particular question explains perfectly the concept of using sanitizers that have an alcohol content of 70% and above. Remember that? It is exactly like that. You'd find that 70% alcohol would remove, okay, let's say this here is like 60% alcohol. You'd still see some bacteria. Then here you have about 40% or 20% alcohol. And in antibiotic 1, it would be like 55% alcohol. You see, the different percentages of alcohol the bacteria or the virus will show different levels of resistance to them. So that's why when they say such, they know they've done the research and understand why you would need to be very strict. Don't cheat the system. If it says 70% alcohol or above, then use that, because that one will be more effective in removing the virus or the bacteria. But if you're going to rely on a cheaper product simply because it's also an antibiotic, you're relying on a cheaper one, it won't be as effective as you want it to be. So actually there's no point in you even buying it. It will show you the results like antibiotic 2 or antibiotic 1. And then question 1.1.6, microorganisms that have a cell wall, no chlorophyll, and reproduce by spores belong to the kingdom of what? Oh, and I see you corrected your answer long before we even marked it. Thank you for that. You see, it's good. You reviewed your answer and then you could see, no, you need to revise it. So which one is here? Microorganisms that have a cell wall, they have no chlorophyll, and they reproduce by spores. What are those microorganisms? Okay. Those ones are A, which is the fungi. Fungi is a cell wall. It has no chlorophyll and it reproduces by spores. It belongs to the kingdom of fungi. Thank you. Next question. Organisms that obtain their food from dead remains of plants or animals are called what? Organisms that obtain their food from dead remains of plants or animals are called what? I see here there's an A. Gosh, I have an A. And there is also a D. Is it, am I following right? It's A and D. Are those your options, A and D? Let me tell you now, before I even show you the answer, it will definitely not be A. When we speak of producers, we are speaking of organisms that actually make their own food. When we say you are a producer, you are making your own food. So it is not A. The answer, yes, it is saprophytes, which is D. These organisms, they obtain their food from dead remains of plants or animals. And then question 8, which group of microorganisms fix nitrogen in the roots of some plants and obtain nutrition in return? Which organisms are those ones? Microorganisms. They are fixing nitrogen in the roots of some plants, and then they also obtain nutrition in them. Yes, now we agree. It is C, bacteria. You have these bacteria, and you'll see them in the nitrogen cycle as well. Ne? They are very prevalent in that nitrogen cycle. They fix nitrogen in the roots of plants. So these plants would usually be roots of legumes, you know. Then they also get some nutrition from that. So it is a, that process is also a, it's actually a, it's a mutual relationship, right? Because they are both benefiting from each other. And then question 9, we're running out of time. Let's finish quickly. We'll finish at least up until question 10. Eh? Question 9, the elimination of undigested food and indigestible material from the body is known as what? Elimination of undigested food and indigestible material from the body is known as what? Okay, both of you, is this the answer? Wait, you haven't answered for this question. No, yeah, I almost went to highlight. What is the answer here? What are what is that process called? Process whereby you eliminate undigested food and indigestible material. What is that process called? Okay, because we are running out of time, I'll quickly give it to you. It is egestion. Yes, Kuzayo, thank you. It is egestion. Think of the E there as exit, excretion. Remember, we even just did the excretion system, the excretory system there. E is exit, so you remove it from the body. You are exiting it. Ingestion, A, is when you are taking it in, right? But egestion is when you are taking it out. Then the last question is, a condition of malnutrition resulting from eating too much of the high energy foods such as carbohydrates and fats is what? Kuzwa, you should know this one. You were very concerned about this one. What is that one here? This condition of malnutrition. When you eat too much of the high energy foods, you see it's even called high energy foods because you would require so much energy to break down this food, like lots and lots of energy. And the answer is obesity. Yo, you see? Obesity over here. You require too much energy, lots and lots of energy to break down these foods, as I've mentioned to you yesterday. So the best way to control your weight is to eat a balanced diet and then couple that with exercising. Because at least when you're exercising, you are making sure you are producing enough energy that can then be there to break down all of these foods. You understand? All right, guys. I'll see you tomorrow. Ne? Don't forget, you can go to Africa 10 Gigs to get your link, and you can, that's where you access your assessment. But please, let's meet together tomorrow here when we do another last round of revision before the assessment. So you'll write the assessment tomorrow, and then Thursday and Friday we will mark the assessment together. All right, guys, thank you so much for your participation. I really love it when we engage, and you guys were wonderful today. Thank you so much. I really appreciate that, and I'm glad to see you are learning and you understand the work. Take care of yourselves. See you tomorrow. Bye.